Welcome to today's webinar. My name is Astrid Gall and I will be talking about Ensembl 96 and Ensembl Genomes 43 releases today. Before we start on that though, I would like to go over some technical things. You will all be able to see that you have a chat box at the bottom in your webinar interface. If you would like to ask questions, please use this chat box for your questions. My colleague Emily will be available and respond to your questions while I'm giving this webinar, and I will join at the end as well. So we will start with a little bit of background to this. I think you will all be aware that we have been sequencing genomes for a long time. The first genome was finished in 1977, and that was a bacteriophage. And the next big milestone was the finishing of the human genome sequence in 2004. And between those two milestones, of course, there were several other species being sequenced, and in some cases, we had later multiple individuals of a species where the genome was sequenced as well, such as for humans. The question is now, why do we need a genome browser? And my answer is this. So a genome is essentially a string of letters, A's, T's, G's, and C's, and we do need a way of making sense of this. And this is where genome browsers come in. They unlock this code of the letters. I work for Ensembl, so I'm going to talk about Ensembl today, but I will not neglect to say that there's also the UCSC genome browser and the Genome Data Viewer by NIH. These tools are all very similar, and it's just up to personal preference which one you would like to use. Now, Ensembl is a very comprehensive resource. We have genomes and gene builds for more than 200 species, and this data is linked to 3 different other types of data. We have variation data, we have comparative genomics data, so that's alignments, gene trees, and homologs, and we also have a regulatory build. In addition to these data types, we have tools. There's a biomart for data export. There's a tool for data processing, the variant effect predictor, which we're going to use later. We also have a functionality where you can use your own data. And finally, if you're so inclined, we offer programmatic access to our databases via APIs as well. For me personally, the best about all of this is that these resources are completely for free, and that includes our whole genome databases on the FTP server, as well as our code, which is on GitHub. This slide shows a phylogenetic tree of all the different species in Ensembl, which are mainly vertebrates, but we also have some model organisms here, which are included as outgroup for this tree and for comparative purposes. So, for example, we have got C. elegans, Drosophila, yeast, and Sayona in here. In addition to this, we also have a sister site, which is called Ensembl Genomes, and this extends Ensembl across the taxonomic space. This sister site, ensemblgenomes.org, is separated into 5 mini sites for bacteria, fungi, metazoa, plants, and protists. This table here summarizes the differences between Ensembl and Ensembl Genomes. Ensembl started 9 years earlier than Ensembl Genomes, but the main difference is really the annotation. For the species in Ensembl, we do the annotation of the genomes ourselves, while for Ensembl Genomes, this is done in collaboration with the scientific communities. So the annotation is provided by somebody else, and we upload and integrate it in our genome browser. Important for today is our release cycle. We try to update our databases as regularly as possible, and at the moment we are working on a release cycle of 3 months. So our last release came out in January, and that was number 95. And at that stage, we have frozen our databases and made several updates to our data and interfaces. So the work behind the scenes that has happened is this. So we added new genomes and new genome assemblies. We made software updates. we updated our gene sets with the new genomes and the new genes. We ran our comparative genomics pipelines, we updated variation data, and finally, we also included a new interface. And then we put out a new release, and this is why you are here today. So at the beginning of April, we have released Ensembl 96. So this brings me to the main part of this webinar, the news in Ensembl 96 and Ensembl Genomes. And this slide is just a summary. So first and foremost, we have hit a mouse genome annotation milestone with the Gencode M21 set. We've also updated the human Gencode gene set to 30. We released a new transcript set, which is based on a collaboration with RefSeq at the NCBI. We made changes to our REST server and our FTP directories. We released 36 new genomes and 6 new assemblies and their annotation. We also included a new interface for configuration of regulation tracks. And finally, we made several updates to variation data and displays. And that's not all. If you want to see all the details, please look at our blog, which you can find under this link here. Okay. So the first big news from this release is that our annotators have completed the manual annotation of the entire mouse reference genome. And this gene set is called Gencode M21. They have been working on this since 2012, and the mouse is only the second species for which this milestone has been reached. The first one, of course, being human. And in addition, they also worked still on the human genome, and this gene set was updated to Gencode 30. In addition to these Gencode gene sets, we also incorporated the so-called main select 0.5 transcripts. So main is a project which is a collaboration between NCBI and EMBL-EBI, and it stands for Matched Annotation from the NCBI and EMBL-EBI. The overall aim is to define a genome-wide transcript set that is matched between RefSeq and Ensembl Gencode, the so-called main transcripts. We are releasing the phase 1 now, which is called main select, and this set consists of one well-supported transcript for every protein coding locus in the human genome. These transcripts are perfectly aligned to GRCH38, and they have 100% identity between the Ensembl and RefSeq transcript. With Ensembl 96, we are releasing the first release. So where can you find this exciting new data now? Well, first and foremost, if you look at the transcript table, you will see that in the last flags column, there's an additional flag for these main transcripts, which is called main select 0.5. So this shows us that our Ensembl transcript here, with the transcript ID ending in 33.6, is the main select transcript. We have also at the same time changed the RefSeq column here. Previously, this just showed any RefSeq associated with our Ensembl transcript. Now, the column is labeled RefSeq match, and it only shows RefSeq transcripts which are in the main select 0.5 set. So these are the ones which are identical to the Ensembl transcripts. So that's one way of accessing our data. There's also a track hub, which you can attach. And I've done this here, and I will show this in the demo as well, where you can visualize the transcript next to your gene sets. And finally, you can also download the main set from the NCBI FTP site. If you want to find out more, again, there's a blog and also a very detailed recorded webinar on YouTube. The next exciting news are that we are in the process of combining the databases for Ensembl and Ensembl Genomes. In this release, we have merged the two REST servers into a single server, which is now rest.ensemble.org. And this meant we had to change the comparative genomics, the genomes, and the info species endpoints. We have also made changes to the Ensembl Genomes FTP directory layout to make it consistent with the directory layout for Ensembl. And these changes affect the variation directories as well as the whole genome alignment files. But the FTP servers are still separate. Furthermore, of course, we have new genomes in this release. There are a total of 36 species in this release which are new, 19 birds, 5 reptiles, and 12 mammals. And this is a whole list with more detail. You can see our birds include, for example, 3 different kiwi species. We have primates, rodents, and other mammals, and we have reptiles as well. We are particularly excited about the fact that we have the genome of Lonesome George, which is this tortoise here from the Galapagos Island, who sadly died in 2012, and he was the last survivor of that species. In addition to the new genomes, we also have new assemblies and annotation. So for vertebrates, we have the koala, a new koala assembly, a new Chinese hamster assembly, and then one for the Northern American deer mouse, and for the duck as well. And accordingly, in plants, we have the kiwi fruit and 2 ecotypes of this grass species here. We had a lot of additional regulation data, in particular coming in release 95 and 93 for human and mouse. The amounts of data for regulatory tracks has increased a lot, and therefore we have developed a new interface that allows you to configure these regulation tracks. At the moment, we have 123 human and 79 mouse cells or tissues, which you can easily choose in this interface. You then need to select the data you would like to see and configure the display in the next step. I will again show this in the demo as well. You can access this matrix from the Regulation tab as well as the Location or Gene tab. Finally, we also made various updates to variation data and to displays. We have included allele frequency data for human from NOMAD version 2.1. We now have variation data for the vervet monkeys as well. For plants, we have variants which come in from the Cereals DB SNP array, which is commonly used in the breeders community for marker-assisted selection. For emma-wheat, we have a polyploid view. And of note for you also is that we have discontinued the variation databases for the fruit fly. Finally, and importantly, we also have made a change to the variant effect predictor. So this allows you now to view the results of your variant effects, on protein structures. And these protein structures come from PDBE. You can not only view the location of the variants, but also the SIFT and polyphen scores, as well as the lost paths for the truncating variants, where the protein is kind of grayed out, as shown in this picture here. Okay, so these are, in summary, the most important highlights, and I will now go and do a little demo to show you this in our browser. So I will leave my presentation, and I will go to Ensembl Homepage. And the first thing I'm going to do is to visit the mouse homepage. So I will click on the mouse here, and then on more information and statistics, where I can find out about the gene sets, for example. And you can see in the table on the right-hand side here that our Gencode version is now M21. And this is the number of coding genes we have annotated. Likewise, if I change the species to human, at the top. And again, click on More Information and Statistics. You can see that we have Gencode 30 now. Okay, I will go back one page using my arrow in the browser. And I will now search for one gene in the human genome, which is LIMD2. And hit enter. I will follow the top hit here to get to the gene type page. And here I'm gonna display the transcript table. Okay, I will make this a little bit smaller so that we can see the whole table. So you can see this gene has 12 transcripts, which are in this table. And the top one with the name 201 at the end is part of the main select 0.5 set. For this transcript, we accordingly also have a refseq match with this refseq accession number starting NM. So let's go to the location tab by clicking on location on the left and add the track hub with the main transcripts. For this, I will go to the configure this page menu. So I click on the button on the left. I'm going to go to personal data and I will do a track hub registry search. And here I will search for main. And Ensembl will tell me it has found it. I will say attach this hub. And then I will configure it. Okay, I will give it a few more seconds. There we go. Now it has loaded. And I click on configure your hub. And I can select how I want to view my tracks. What I'm going to do is I will add the labels onto this track as well. So here you can see the track hub is selected, and I will click on the box next to it to say I would like to see the structures with labels. And I will close this menu and it will load the track onto my region and detail view. LIMD2 is a gene with transcripts encoded on the reverse strand, as you can already see. So we are going to look for the main select transcripts on the reverse strand as well. You can see there's one at the top here, which is a forward strand. And there's a track here, main select 0.5 on the reverse strand. To be able to view this a bit better, I will grab this track and move it next to my gene track. And here you can see how our golden transcripts lines up perfectly with the reference sequence from NCBI here. And the second transcript shown in the main select track is our Ensembl transcript shown in red. What you can also do is you can expand this track here, human cDNA, refseq, and ENA, and show labels on here as well. So I click on that, and then the cogwheel, and I say I want to see labels. It had to think for a moment here. Okay, and this is now expanded into the different sequences of the cDNAs. And you can see here again is my refseq sequence, which matches our main select transcripts. Okay, so what I'm going to do now is I will remove this track hub now by clicking on configure this page and then personal data. And then I will use this bin icon here next to the name of my track hub. Likewise, I will go and to configure region image and reset my configuration of my view here. This is so that we can start with a fresh page and start configuring some regulation tracks in the new interface that we have introduced in this release. This is very slow, I'm afraid. Okay, I will close this and I will go to the new interface directly. Okay, to find this new interface for configuring regulation tracks, again, you go to configure this page. And then in the regulation section, in our regulation section, there's a new item here which is called features by cell type. So I will go there and click on that. Here you can now see the two steps that you need to do. In the first one, you need to select your tracks, which is where we start with our cell lines and tissues. You can do this by alphabet. You can select everything, and you can also search for one cell line. And I will search for HepG2 and select that. And then you need to select in the second tab the data that you would like to see under experiments. Here you have options for histone modifications, where I will select this one here, H3K4ME3. And I will also select some transcription factors, namely this one here, REST and RAD21. I will not select anything for open chromatin. So now that I've selected my cells and what data I would like to see, I can click on Configure Track Display, which takes me to the second step. And here you can see a little matrix of the cells and the experiments, and you can click on the cells in this matrix and then select if you want to see peaks, signal, or both. And you can also select or deselect columns, rows, and individual cells. So I will select everything here. And here on the right-hand side, there's a summary what you have selected. So you can expand these things. And I will now click on View Tracks. My data is loading onto the third picture on this page here in my region in detail view. Yeah, I'm afraid this is a little bit slow now. I have not selected a lot of data. Now it has done it. So if you scroll to the bottom of the page now, you see there's an additional track here which is called HepG2 in light blue. And you can see data from the experiments that I have selected. So for example, DNA methylation right here, and then predicted transcription factor here, here, and here. And I can see both raw data signals at the bottom, which were used to annotate the peaks, which are the colored blocks here at the top. Okay, so that's what I wanted to show here. Now I will go back to the homepage and visit my full list of Ensembl species here. So this is a table with all the species that you have. You can download it in Excel format, and you can also filter it for species such as kiwi. But if you want to know what's new, the best way is to go to the table of assemblies here. And this shows you all the different releases and assemblies we had in Ensembl. Here at the beginning, release 96, and you can see in dark yellow the new assemblies we included, including the one of the tortoise here. Okay, finally in Ensembl, I wanted to show you the web and the new protein structure view in the web. So I'm going to go to the web tool in the bar at the top and then use the web interface. I have run a job before. If you haven't, your page will look like this. So we give this job a name. I will call it live webinar. And then we put a variant on chromosome 19 at this position, which I'm now typing, 39731447. The variant doesn't have a name, and it's an A to G change. And we need 3 more dots because it's a VCF formatted variant. I'm not going to change much here. I will just go into this section here, additional annotations, where you can find one tick box for protein annotations. And if you tick this, as it says here, you will see where available the variants on a PDE protein model. So tick that, and then click on Run at the bottom of the page. At the moment, our jobs are here summarized in this table, and while it's running, it says queued. If it's done, it turns green and you can click on View Results. So these are the results for my live webinar now, so I will click on those. And I will show you where you can find this column with the protein structure view in the output table. At the top, we've got a graphical summary and a table summary for our variants. And then here in our detailed web output table, there is a column towards the very right, which is called domains. And there we have a button, protein structure view. So I click on that. And this opens our light mole protein viewer. So this is an interactive view of the protein. You can see the variant here, which is a valine to alanine change. You can highlight your exons in different colors by clicking on this button on the eye icon in the menu on the right. You can do this with PFAM domains as well. And finally, you could also highlight SIFT and likewise polyphen scores. Okay, so that's what I wanted to show in Ensembl. I will now go to Ensembl Genomes briefly. This is the homepage. As I said, we are on release 43 now. The main news are in Ensembl Plants. So I'm going to go to Ensembl Plants. Here at the top right, we have the summaries about the news in Ensembl Plants, so the new genomes, our markers for wheat from the cereals DB array, and the polyploid view in ML wheat. I'm going to go to click on wheat here, the Latin name, and just go to this example gene here. And follow the link to the gene ID. So I'm now in the gene tab for this gene of wheat, and I will show you where you can find these new variants. So you could go to the variant table here on the left, and this table summarizes all the variants in this gene. You can filter this table by source. So if I filter by source, I can see there are 23 variants from cereals DB. I will switch the other ones off and click apply. You can see that you have applied a filter by this green X here. And now, these are the variants which are from this array, which is important for breeders. Some of them are missense variants, and they do have SIFT scores. Okay, so that's all I wanted to show in my demo. I will now go back into my presentation for a little bit of a summary. I hope you found this helpful. If you would like to learn more about Ensembl, you could go to the Train Online by the EBI. So we are part of EBI Train Online, and you can find our courses there. We also have a dedicated site with tutorials, which you can find here. I've already mentioned our YouTube channel. If you are ever in China, this is also accessible from Youku. If you have any questions, comments, or suggestions about Ensembl, you can email us at helpdesk@ensembl.org. You could also join one of our public mailing lists. There's a developers mailing list, dev, where developers ask questions about programming and large-scale data access. And there's an announce mailing list where we talk about our new releases, new interfaces, new data that we have and that we're planning to include. If you like what you have seen, you could also ask us to host an Ensembl workshop. We can teach these courses for free at your institution if you are in an academic institution in a low and middle income country. If you are in a high income country, you will have to pay for the trainer's expenses. We do browser courses, which are between 1.5 and 2 days. And these are aimed at wet lab scientists. We also have a REST API course aimed at bioinformaticians, which is a bit more than half a day. And finally, we have a Train the Trainer course as well, specifically for Ensembl. If you're interested in hosting a workshop, again, please get in touch at helpdesk@ensembl.org. You can also follow us on social media. We have a Facebook account, we have Twitter accounts, and we have a blog. If you use our resources, please cite us. This is the list of our publication linked at the top there. It's very important for our continuous funding that we get citations, and we do provide this resource for free. And this is us last summer. Most of these people are database managers, bioinformaticians, programmers, and so on. And it's just the pleasure of the outreach team to do training like this and talk about all the hard work that the other people are doing. And this brings me to the acknowledgement. So I'd like to thank the entire Ensembl team, which is listed here, and of course our funders. Our main funding comes from the Wellcome Trust, and we get a lot of co-funding from Ensembl, from the European Union, and other funding agencies. So thank you very much for attending.